Okay. So, hello everyone. My name is Ankita. I am your TA for this course. We will be having one live interaction session every week and around this time from 4 to 6. So, basically, the idea of the sessions is to discuss any problems that any of the students, any of the course attendants, anybody is facing. And apart from your questions, I will also be going through a recap of the topics that have been discussed in the past week and a few questions that were part of the previous year assignments so that you can get an idea of how to solve them. And apart from that, once the deadline for the assignment for that week is closed, we will discuss those questions as well. And any ideas or anything that you wish to discuss, we can come up with those as well. And please give your suggestions as well, what you would like the live interaction session to be, if you want it to be different in any way, or if you need any other sort of help, for example, some book recommendations or different types of questions, so we can modify those accordingly. So as of now, the presentation that I have for this week is basically involving a recap of what were the topics that were discussed, and then we'll move on to some sample questions based on previous year assignments for NEPTIL, and hopefully that will help you solve your assignment for this week as well. Please interrupt me at any point of time if you have any doubts. I will try my best to solve those as well. So for the course, Cell Biology, Cellular Organization, Division, and Processes. The first few lectures, the first four lectures instead, have basically given a comprehensive idea about what a cell is, what are its different components, what are the basic processes which are involved in the essential, which are survival essential for a cell, which include transcription and translation, what are the different compartments that are operating together to help a particular process along, and so on and so forth. So the first idea that was discussed in the lectures was about the discovery of a cell. And as you remember, it was about how Robert Hooke observed these brick-like structures under the microscope for the first time, and he defined it as the structural and functional unit of life. Not so strongly, but that was the first observation that was made by Robert Hooke. And if you can see the figure on the right, you'll be able to see a sample of the kind of microscope that was used and the first observation that was made by him as well. I will be sharing these presentations as well. If you want to look up the references, you can surely go ahead and do that. If at any point of time I'm not audible, also please let me know. And again, if any questions at any point of time, you can go ahead with those as well. So yeah, so the first idea was by Robert Hooke, and then these were further explored by Anton von Leeuwenhoek. These are all the initial scientists, the initial discoveries made by some prominent scientists that actually led to the field of microbiology and what we understand today about cells in the long run. So Leeuwenhoek had observed Spirogyra and he named them as animalcules, which meant little animals. And he also looked at blood cells from several animals as well. Then came Schleiden and Schwann. These two gave rise to the idea of the cell theory. And they had stated two major ideas on their behalf. That first was that all organisms are made up of one or more cells, and that cell would in fact be the basic unit of life in all sort of living things. So this was the basic idea of the cell theory, and also that cells are produced from pre-existing cells. They cannot be formed ab initio, and that was the idea that was brought forth by Robert Virchow in 1858. And what you see on the right is again the Leeuwenhoek microscope that was used. As you can obviously see, these are very, very simple microscopes that were, we call them simple compared to the kind of technology that is used today, how advanced and how complex those are. But obviously the idea remains, the basic idea remains the same of magnifying a very tiny object. And back in those days, without any extensive knowledge about topics like we have today, these were exceptional discoveries. And what you see on the right is also an original snippet of the kind of section that Leeuwenhoek had observed. And you can see that there's a section of a spinal nerve which he had seen under his microscope at that point of time. Then came, oh, sorry. Then came the idea of the modern cell theory, and it talked about how cells are the fundamental and fundamental structure and function unit of all living beings, how organisms are made up of one or more cells, which can arise only by pre-existing cells, and they carry the hereditary information which is being transmitted across generations, and how they basically have the similar chemical composition and the structural organization. When we say this, it's a very generic statement. It will, of course, depend on the type of cell, where it is present, and what sort of organism it is present. There would be subtle differences in this regard, but the basic idea is the same, where you have an outer envelope which is made up of lipids, and you have a cytosol that comprises all of the different organelles. There would be increased complexity and decreased complexity depending on where that particular organism is in the animal kingdom, not animal kingdom, but in the living world. But the overall idea remains the same. Um, then, as you can see here, you can see that there are different types of microbes that have been shown in the lower figure. I don't have a laser pointer, but yeah, you can observe it. So first is E. coli, Escherichia coli, and you can see that there are two different types that have been mentioned here: gram positive and gram negative. If you've gone through the lectures, you would have seen that the major difference between them is based on their envelope, or based on their outer membrane. The outer membrane, the peptidoglycan layer, and the type of composition which is present in the outer membrane and the peptidoglycan layer and the cell membrane of these type of organisms leads to differential staining of these organisms by a particular type of stain, which is known as gram stain. And depending on whether or not they get stained, they can be classified into two different types: gram positive and gram negative. Mostly gram negative bacteria, majorly, it's not a general rule, but majorly gram negative bacteria are believed to be pathogenic, and the diseases they cause are extremely severe, and they are, when I say severe, they are treatable, but they're majorly associated with pathogenic diseases. And then again, you have cyanobacteria, which are uh, a bit of a misnomer, but they're mostly known as blue-green algae because they're not actually algae, but this is their common name. But the reason this name has been attributed to them is because of the presence of chloroplasts, because of which they are able to carry out photosynthesis. So that is another subclass under these organisms. Now you see the differences between prokaryotes and eukaryotes. Obviously, the unicellularity and multicellularity is is some issue here. Okay. The unicellularity and multicellularity is a major difference. The size range is quite varied. The nucleus or nucleoid-like structure vary between the two. DNA circular or linear. Cytoplasm has ribosomes, or it has ribosomes along with membrane-bound organelles. The presence and absence of mitochondria, and the presence of or absence of centrosomes. These are all of the different characteristics that vary between prokaryotes and eukaryotes. Then came the idea of the endosymbiotic theory, and this is believed to be one of the preliminary ideas that came about as to how prokaryotes, how from initial prokaryotes, the modern eukaryotes have come forth. And the basic idea that was explored in this regard involved, I hope it is visible, I think it's a bit small. Just give me a second. Okay, so the basic idea was that there was a preliminary organism which was a sort of an ancestral eukaryote, and this was anaerobic in nature, and the kind of metabolism that this organism had was believed to be insufficient, or was not capable of adjusting to the environment that it was surrounded in. And that was believed because the fuel that it was, or the food that it was taking up, was not getting completely utilized or not getting completely digested or metabolized. So for this reason, it is believed that an aerobic organism, which is of course a bacteria in this case, was engulfed by this ancestral organism, and this eukaryote would now be able to sustain the life of this now newly engulfed bacterium, and that would duplicate or replicate inside the ancestral eukaryote, independent of any other external resources, and therefore it forms a symbiotic relationship where both the organisms are able to benefit from each other, and that equation is helping them survive individually, and therefore they would support it over the long run. So now this symbiotic system is allowing it to carry out the aerobic catabolism, and some bacterial genes would also be utilized by the eukaryote, and some of the advantages would also be taken up by the other organism as well. And eventually what it is believed is that this aerobic microbe or this aerobic bacteria eventually evolved to become what we know today as the mitochondria because of its aerobic nature. On the other hand, this actually led to the formation of the non-photosynthetic eukaryotes. But on the other hand, plant cells or any photosynthetic organism that contains chloroplast is believed to have evolved because it engulfed a photosynthetic cyanobacterium, and this was also able to independently function and was able to provide the ability to utilize light as a food source and then produce CO2 from the same. So this photosynthetic eukaryote would now be capable of evolving eventually into the plant cells. What you need to keep in mind is that this entire series of events that is being described is believed to have occurred over thousands and millions of years. It is not something that happened in a very short point of time, and therefore there must be several like infinitely huge number of stages where organisms have evolved over generations, and where some organisms have only been allowed to sustain while others were not, and these were believed to have happened because they gave a survival advantage to the initial eukaryote, which is why it has sustained and evolved in this direction. Uh, then came the idea of the type of chemical bonds that was explored. So we talked about ionic bonds, about covalent bonds, metallic bonds, hydrogen bonds, etc. Each of them involve different types of interactions. Like, for example, covalent bonds involve the sharing of electrons, which we know is one of the strongest bonds that exist. They can be polar or apolar in nature. You have ionic bonds where one atom is giving up its electron and the other one is gaining or receiving that electron, and therefore that is the strongest as well. Then comes the metallic bond where positive metal ions are attracting each other, and you have hydrogen bonds, which is definitely not a strong bond, and you can see that it involves an electronegative atom, and you can see that there is a sort of a dipole sort of interaction which is occurring, that there is a shift of the electron sharing towards the more electronegative atom, while the other one is deprived of the same. And this interaction, despite the weak nature, is extensively present in the biological system. And finally, we have van der Waals systems as well. So these are all the different types of interactions that you can often see in living organisms. Then came the idea of the organization or the molecular organization that exists. Again, I am not able to see the screen at all points of time. If you have any doubt, not necessarily related to this part, even from the topics that come up later, please do interrupt me, and I'll try to help you out to the best of my abilities in that regard as well. So talking about the molecular organization of cells, if you have, I'm sure you must have come across it at some point of time before, but if we start from cells, the entire living organism is made up of cells which become tissues, which tissues are ending up forming some organs form organ systems, and organ systems are leading up to the entire living organism. However, within the cell, there is also extensive hierarchy of organization, and there are a lot of checkpoints that are in place that are making sure that each of those systems are functioning properly in order to allow the cell to survive properly at large. So, for example, if we see the different types of organelles, among them you can see the monomeric units of nucleotides are forming DNA, not just DNA, even RNA as well. But with respect to DNA, this is able to form chromosomes. Chromosomes is forming the entire chromatin, the entire genetic content of a cell, which is residing within the nucleus. Then you have amino acids, which are forming proteins, polypeptides, and then proteins, which also have several different levels of organization. And then these proteins are getting transported to different regions within the cell, and they are capable of performing their independent functions. Now, proteins, while DNA is the genetic material of the cell, and it is extremely crucial because it is what is carrying information from one generation to another, just having DNA inside the cell will never suffice. You need a molecule that is able to execute the functions that the DNA is dictating. So it's like having just a person having a brain will not be able to move, but a person with a brain and a functioning locomotor system would be able to move from one place to another. Like a similar analogy in this regard as well. So these proteins are then getting inserted in different regions, like for example, the plasma membrane, or they are being secreted, or they are performing some intracellular function by itself. So these are the different types of jobs that they execute. And you also have sugars, which are forming structures like cellulose fibrils, which are extremely crucial and are involved in the formation of the protective structure around several cells, mostly plant cells, which is the formation of a cell wall. So you can see that each of these macromolecules is made up of very simple subunits. All the examples that I cited just before this are in that regard. Then you have the organization of proteins. So we looked at the basic amino acids. Just a second. We looked at basic amino acids being able to form these primary structures, which are joined in a line, and they form a polypeptide, which polypeptide eventually forms secondary structures like alpha helices and beta sheets. Then you have the tertiary structure, which involves a 3D conformation of these independent secondary structures, and ultimately motifs and domains, which are leading to the formation of a quaternary structure where different subunits are coming together and helping the protein execute its physiological function. Because we were talking about protein, so in that regard, the next type of or the subtype of proteins that are involved in a certain type of function are enzymes and coenzymes. So what are enzymes? Enzymes are biological catalysts, and what they basically do is that they are responsible for increasing the rate at which, or rather, if we frame it more accurately, then they are involved in decreasing the activation energy that is required to perform the reaction that is converting the substrate to the product. So if you have substrates at one level, and you have like if we consider an energy graph, then if you have substrates at one level and you have the product at another level, then the reaction has to proceed through a sort of a curve where there is a certain amount of activation energy that is required to tip the scale in the favor of product formation rather than the reactants staying as it is. And this energy that is required, that is being reduced by these biological catalysts such as enzymes. So while enzymology is a very extensive field where there's till date, of course, a huge amount of research is being carried out, you can read about it a little bit in detail if you want. You can refer to Leninger, Principles of Biochemistry by Albert Leninger. It's one of the finest books where you will get the basic idea regarding enzymology and all of the different equations that are involved, the basic idea, the lock and key hypothesis, the current theories that are coming up in this regard. You'll get a very nice comprehensive view about enzymes as well. So coming back to the slide again, so like you know that enzymes are commonly proteins, and they bind substrates in a way that ends up lowering the activation energy of this reaction, like I mentioned. And they usually have much higher affinity for these transition states, which are usually considered unstable, than the substrate native form. And what that effectively does is it lowers the energy, the activation energy, and therefore it favors the transition state over the substrates that were initially present. And they would therefore tip the reaction in favor of formation of the product rather than reverting back to the substrates. So this is the basic job of enzymes, and they are also believed to be very, very specific. When I say specific, it means that for a certain type of a reaction, only a certain type of enzyme can usually perform the job. You wouldn't find a large number of enzymes that are very non-specific in their nature. This is particularly put forth by the cell in order to maintain how do I put it? In order to maintain the accuracy of the way in which reactions are being carried out in the cell, so that you wouldn't just any part of the cell is not just executing any function, and there is just to avoid anarchy. Let's put it that way. So that is the job of enzymes. Then come coenzymes. So coenzymes, I believe they are not protein molecules. First of all, they're quite small, and they're organic molecules, and they work in conjunction with the enzymes, and they further enhance the reaction rate. So at the physiological level, where the rate at which reactions occur are way faster than what we are used to when we are thinking about chemical reactions in the laboratory or somewhere else. So in this case, the presence of coenzymes would many folds increase the rate of that conversion, and these molecules are often extremely crucial. You'll find a large number of enzymes that would ideally not be able to perform the reaction that they normally do without the presence of coenzymes and cofactors as well. They're very, very important. Like if you have come across EDTA, for example, which is believed to be a chelator, it is often added in a like, for example, if you think about blood vacutainers, if you have ever gotten yourself tested for like any pathology lab, basically, they usually collect your blood in these little vials. Glass vials, and these are called as EDTA vacutainers. So they contain the inside of those vials are covered or coated with EDTA, which is a chelator. And what it does is that it chelates divalent cations. And what that effectively does is it makes them unavailable for any digestive enzymes which might be present either in the environment or in in the system or somewhere in the vial, just to prevent any digestion of any nucleases or any essential component within the cell. So that your blood, it remains, it does not coagulate, and it remains fine by the time it reaches the pathology lab, and it can be used for whatever assays are necessary. This was just one very common example. If you have worked in any sort of a research lab or have been a part of it during your education, you would also see the extensive use of such chelators like EDTA and EGTA, because they are very, very crucial in preventing those enzymes from working and essentially maintaining the integrity of whatever biomacromolecule you are currently interested in. So those were cofactors that I talked about, the divalent cations. When we talk about coenzymes, coming back to the slide again, these are small organic molecules, and they enhance the reaction rates, like I said, and they are recycled to participate in multiple enzymatic reactions. And this is again a major point of difference between enzymes and coenzymes, because like enzymes are extremely specific, they can't just haphazardly work in any reaction. Coenzymes, on the other hand, can be managed across different sort of reactions. For example, nicotinamide adenine dinucleotide, which was NAD+, is an electron carrier in oxidation reduction reactions, otherwise known as redox reactions. And NAD+ effectively accepts two electrons and an H plus ion from one substrate, and it forms NADH. Now, NADH is much more stable, and it is capable of donating two electrons to another substrate, and it can again form NAD+. So it is responsible for this sort of transport or shipping of electrons from one place to another. So it's an electron carrier in that sense. So these coenzymes are involved in such kind of factors. Somebody's trying to join. So after that, so the basic idea of the cell and what the evolution of prokaryotic cells have been, the differences between those, and the basic units inside a cell or inside any chemical entity were discussed. And after that, we come to a little more detail about cells themselves. So what you can see here as images are, the first one is actually showing the different components. I think I'll zoom a little bit. Yeah, the different components of a cell. And if you look here, of course, on the outside is the plasma membrane, which has been magnified here, but it is basically this layer. And you're already familiar with it. It is a lipid bilayer, which we will again discuss in quite a lot of detail and has been discussed in the lectures as well. It is the most, while it is usually not considered as an organelle, it is extremely crucial because it is what maintains the difference between what is cell interior and cell exterior. So that is its major job. And of course, apart from that, it performs a number of functions, which we'll again come to in a little while. Apart from this, when we talk about all of the different organelles, when we think of a cell, the first thing that you would ideally be able to observe under any microscope would be the nucleus, which is usually a very dense, dark colored sort of structure right in the center of the cell. And the nucleus by itself um, would not be very useful. It is useful when you consider a cell at large. But why is it given so much weightage? Of course, because it carries the genetic information inside a cell. So at the time of division, when a cell divides from 1 to 2 into the daughter cells, the one most important factor, of course, all the others are important as well, but the one major important factor is that the information that is within the nucleus should undergo correct, accurate division into two, first duplication into two, and then equal division into the two daughter cells. Any sort of anomaly in this, any deviation from this accurate distribution of genetic information leads to a number of diseases. Um, Of course, the first thing that comes to mind whenever we talk about incorrect cell division is cancer. Now, that is not the only reason that cancer arises. A number of mutations have to come into play, and a whole large number of things have to go wrong for a cell to actually convert into a cancerous or a neoplastic cells. There are a number of steps that occur before that, but yes, one of the major things that does occur upon transformation to neoplastic condition is that the nucleus becomes aberrant and the division is aberrant, and it leads to a large number of problems. So that is what we see here. Now, the nucleus, like I said, contains this hereditary information. And if you think about it, then uh, if you have come across genes before, you might have heard of, the human genome and it being sequenced. And that was a huge project, and human genome project, basically, that was taken up by a large number of institutes or laboratories across the world. And they decided that they would sequence the genome, which is indispensable information in today's date, considering how much work is being done based on genomics, so much of diagnosis, treatment, understanding genome prognostic, which means that you are trying to understand what is the possibility that a certain person can develop a certain disease based on just their genome sequence. So all of these studies, all of these research areas, they have opened up after the sequencing initiative has been taken. So all of this information that we are struggling to even store in today's day, how is it inside the cell? How is it being managed or accommodated inside a cell? So that in itself is another area of study, which again, while it has been understood to a large extent, there is still a huge amount of research that is going on in that regard as well. But if I just sum it up a little quickly, then it is basically there is heterochromatinization. There's heterochromatin there, and there is euchromatin inside the cell. So there is extensive packaging of DNA that occurs inside the cell. So you have a nucleic acid and you have certain proteins which are commonly known as histone proteins. Now, these histone proteins, I always use this example because I feel it's the easiest to understand. Use a wired mouse. So imagine this is your histone protein, and the wire that comes with it is your DNA sequence. So it basically wraps this DNA. You would do this without even realizing it consciously, right? That you keep your mouse like this when you want to save space, or you keep your earphones wrapped around something like we used to before they became wireless. But whenever you want to package something, you would fold it and you would, make it as compact as possible. The cell uses the same strategy. So it packs all that information that is present in the DNA into reasonably small, into an extremely small, not even reasonably small, an extremely small space, which is just the nucleus, by carrying out extensive packaging. So that is why you see DNA in the form of chromosomes. And those chromosomes all together are being packaged inside the nucleus by a huge number of interactions. So that in itself is a very interesting area of research if anybody is interested, and you can look it up. So coming back to where we were discussing, so the nucleus, like you can see, has been indicated at the center. It is a dark colored or an intensely colored structure inside the cell, which has some demarcation within itself as well. It has the nucleus and the nucleolus and the nucleoplasm. Um, while these are not very clear membrane bound regions, you can see a difference between them. There is a difference in the type of in the composition of these regions based on which the segregation has at all been decided. And the nuclear membrane, if you see, you will see that it is not continuous. You will find little tiny gaps that are present, and those are known as the nuclear pores. So those pores are the regions through which some transport is being carried out, and we'll come back to it when you must already know about it, but it is basically involved in transporting a large number of things. But one of the major things that is involved in transport is for mRNA, right? So anything that is being synthesized inside the nucleus, if it has to move to the cytoplasm or either way around, for that you need to have some regions of accessibility, and those are the nuclear pores. So right around the nucleus, you are seeing these sort of crinkly structures that are present, and these crinkly, it's not just a structure. In fact, it's a network. It's quite extensive. It's like a mat that you see, and that is of course the endoplasmic reticulum. The reason that it is appearing a little sort of spiky or crinkly in nature is because that is the rough endoplasmic reticulum, which houses the or which is because of the presence of ribosomes on the surface. And ribosomes, as was discussed in the lectures as well, and you might already know, is the major molecular machinery, or in fact, it is the molecular machinery which is absolutely crucial for the synthesis of proteins. So these are, if you just think about it, then you'll see that this explains why all of these structures are so close to each other. It is because whatever DNA duplication is occurring in the nucleus. After DNA has been duplicated, it needs to undergo transcription, which is that the information that is present in the gene needs to be converted into something which can now be read by the cell further. And that conversion occurs into the mRNA. This mRNA formation, or rather the process of transcription, is also occurring inside the nucleus. Once the mRNA has been obtained, it needs to be shifted, or it needs to be moved or transported outside of the nucleus into the cytoplasm, because that is where you see that the ribosomes are present. After they have been translated from the mRNA into the protein, into the polypeptide that it needs to be functioning, that polypeptide needs to undergo folding. Now, folding and conformation are extremely crucial, because just a simple primary structure or a straight linear structure of a polypeptide is not going to be able to do anything. It has to fold, it has to go through all of those stages. It needs to form secondary, tertiary, and eventually quaternary structures, and needs to form the entire protein at large for it to be able to function. We'll see one example of it, which is F1-FO-ATP synthase, which performs beautifully, and you can find such wonderful animations of it online, because so much has now been studied regarding the structure of proteins, especially after cryo-EM has been understood, cryo-electron microscopy has been understood, where you can really understand the dynamic nature with which proteins function. So these are all brilliant techniques that are coming up in the recent past. So we talked about the ER. Now, that is not the only type of endoplasmic reticulum. The ones which look crinkly and which are right next to the nucleus are the RER, or the rough endoplasmic reticulum. But you can also see some relatively smoother structures which are much farther, and that is the smooth endoplasmic reticulum. Now, don't get fooled by the similarity of their names. Their functions are extremely different. It's just that they are similar in structures, and because ribosomes can assemble and disassemble, but the functioning of these compartments is carried out separately. Apart from this, you also see peroxisomes, which are tiny structures which are present. There's tiny vesicle-like structures which are present inside cells, and they are involved in managing the reactive oxygen, reactive nitrogen species, which are being synthesized inside the cell for processing of the same. You also see vacuoles, which are tiny empty sort of structures which are present inside the cell. You see a very small one here because it's the representation of an animal cell. If you see plant cells, their vacuoles are huge. There is a central vacuole right at the center. And in that case, its job is to provide, apart from its normal function for, maintaining the ionic balance, etc. It also is responsible for maintaining the turgor pressure inside the cell, which is very crucial for plant cells because it gives them the mechanical strength and the support that is needed to survive in whatever environment they might be in. Then you also see centrosome, which does not normally have much of a role to play in a non-dividing cell, but at the time of division, it is, if not anything, if not the most crucial, one of the most crucial roles to play at the time of division, because that is where some structures known as microtubules or the spindle fibers, they attach and they divide the cell into two. So it has a very crucial role at the time of division. You also see microtubules, which we'll come to soon, and lysosomes as well, which are considered to be the digestive bags of cells, and they are involved in chewing up whatever food is being taken up by cells. You also see Golgi apparatus or Golgi vesicles, which have a separate type of function involved also in vesicular transport and processing. And whatever remains apart from all of these structures, that is basically your cytosol. Now, when we think about such images, it appears as if the cytosol is just a background, right? It looks like a clear background within which each of these wonderful organelles are present, and therefore the rest of the cell appears to be empty. But of course, that is not the case. If the cell had so much space to spare, why wouldn't it just become a smaller structure? That is because everything that is placed inside the cell is placed on the basis of some railway tracks which are inside the cell. Now, these railway tracks, or otherwise, more scientifically speaking, these structures which are believed to be the cytoskeleton, are extremely crucial for the proper organization and proper transport inside the cell. The cytoskeleton, like if we go by the name literally, means the skeleton of the cell, right? Skeleton means something which appears like a framework, and over that framework you can transport something. That's why I mentioned the analogy of like a railway track. So the image that is, sorry, yeah, the image that you see right next, this beautiful, beautiful image, it's a confocal image. This is the representation of what actually is happening inside the cell. So like I said, there is not just a clear background. It's not an empty space. It is completely filled by all of these string-like or fiber-like structures, which is known as the cytoskeleton. So if you go by the color, the structure that you see blue and intensely blue in the center is, if you might not have guessed already, of course, it is the nucleus. The most commonly used stains are DAPI, DAPI, you can look them up. And there's another stain known as Hugst. These two are commonly used to stain the nuclei, and they're very common stains. Now, when I say stain, they basically, they're phytochromes, which are used for immunofluorescence imaging, and that helps you to uh, discriminate or to be able to distinguish between different areas of a cell. Otherwise, you would never be able to see, right, what is happening. If you want to know if a particular protein is in the nucleus or not, then you need a nuclear stain. There are such stains available for each compartment. For mitochondria, you have for Golgi, etc. So if you want to label one particular region and you want to see if your protein is expressed there or not expressed there, you can perform such assays to distinguish between them. So this is a it's an imaging technique basically. So the blue structure is your nucleus, and what you see on the boundary of the cell, like there are lots of cells here, of course, but what you see at the boundary is actin, which is a component of the cytoskeleton, and actin is majorly present right next to the cell membrane, and therefore it is very commonly used to demarcate the cell boundary. What you see in the interior is the extensive network or mesh work, even you can call it, of microtubules, which are also a type of the cytoskeleton. So I'm not sure if this course has it, most probably doesn't, but if you want to read up on microtubules and actin, as well as another type of cytoskeletal element, which is the intermediate filament, you will find beautiful descriptions of each of these in books which are written by Cooper and Karp. These are very commonly used cell biology books, and they have such beautiful descriptions. You can certainly go through it. Or in case if you want more details, we can discuss that in the next class. I'll include a few slides on them. So yeah. So this was what I talked about the cell. Now we have talked about the organelles which are present. We have talked about the cytoskeleton. What remains to be discussed in this regard is the composition of the plasma membrane that I talked about initially, that it looks like a lipid bilayer. Now, when I say lipid bilayer, what is it? What are these lipids? So here you see the different types of membrane lipids which are present. The most common, or the majority of any membrane that we know about, any biological membrane that we know about, are phospholipids. Now, phospholipids, as the name indicates, they have a phosphate group, and the remaining of it is the lipid moiety. So it has two hydrophobic fatty acid chains that are linked to glycerol, and the third carbon atom is bound to the phosphate group, hence giving rise to the name phospholipid. And this, in fact, this phosphate group can be linked to some other sort of moiety, some other polar moiety, like choline, serine, ethanolamine, or inositol. So you have phosphatidyl choline, phosphatidyl serine, phosphatidyl ethanolamine, and phosphatidyl inositol. each of these have very, very specific functions, and the presence or the ratio at which they are present can actually alter the function of that cell. Or let me give you one example: phosphatidylserine. The amount of phosphatidylserine that is ideally present on the surface of the cell is very, very tightly controlled. And if there is a shift in that, very tightly controlled, when I say, I mean that if you think about the lipid bilayer, which has two layers, right? The presence of phosphatidylserine is mainly on the inner layer. If this is cell exterior, this is the cytoplasm, and this is your lipid bilayer. You will find on this surface that there's the presence of phosphatidylserine. But when a cell is in a very stressful condition, it doesn't have food, or it is seen that it is somehow its functioning has been compromised, then under those circumstances, the cell can shift its a large percentage of its phosphatidylserine towards the surface. This shift, which seems like a very small change to actually occur, is a huge how to put it, a huge signal for the surrounding environment. It acts as an eat me signal. Again, this is something you'll find in cell biology books very extensively, that the presence of phosphatidylserine on the outer leaflet of the lipid bilayer is considered to be an eat me signal, that the cell is asking its environment that I am not healthy, you should eat me up, and you should use my resources for the sustenance of other cells, for the sustenance of the organism at large. So the membrane composition, or the percentage of membrane, percentage lipid composition of any cell membrane, is also extremely crucial. So you can see that the percentage of these lipids has also been mentioned in each regard. Like for phosphatidylcholine, it's 17% in RBCs, phosphatidylethanolamine is 18%, and so on and so forth. The second type of membrane lipids are glycolipids. Again, the name distinction is indicative. It means that it has two hydrocarbon chains and it is linked to a polar group that contains carbohydrates. Third are cholesterols. These are not similar to this. They do not have a similar amphipathic structure, but instead they are much more rigid. It has four hydrocarbon rings that are very hydrophobic in nature, and one end of it has a small OH group that is very weakly hydrophilic, and the presence of cholesterol in the membrane can significantly alter how stiff or how relaxed that membrane is. And in fact, this also will vary from this will vary across organisms that live in different environments. You can again read up on this in details. You'll find it in these books as well, in even in Leninger or any of the common cell biology reference books that you want to look at. So yeah, so depending on whether an organism normally lives in a very cold environment, or a relative, or lives in the plains, or lives somewhere close to the equator, the lipid composition of the same also varies. So again, it's something that nature always helps, that it helps or allows for survival of the fittest, and evolutionarily this has been seen as a useful adaptation. Uh, Next comes the models of the plasma. Come the models of the plasma membrane. So we talked about how the membrane is very extensively rich in lipids and in different types of lipids. When we say types, we mean the different compositions of, or the different percentages of each of these phospholipids, and the subtypes of phospholipids, and so on and so forth. So if you see this representative image of the phospholipid on the top, you will see that it has a hydrophilic head and it has two hydrophobic fatty acid chains. Now, this orientation is extremely important because that is what gives rise to that lipid bilayer structure. It's because the hydrophobic side will always shy away from the surrounding polar environment, while the hydrophilic side, which are those tiny bulb-like head structures that you see, will always be facing the surrounding environment. So in order to keep those hydrophobic sides on the inside, the membrane arranges itself in this way such that it is, you can never cut through a plasma membrane and expose it. It will always try to cover itself, and it will form a closed structure such that the hydrophobic sides are always on the inside. It is the nature, or it is rather the chemistry of a membrane that allows it to survive in an apolar or aqueous environment around itself. So among these, you can see like even in the representation, you can see that the outer two sides, the yellow colored hydrophilic heads, are believed to be the polar entities, while the little hydrophobic tails that you can see at the center are non-polar in nature. Apart from this, you can also see that there are several different types of proteins that have been shown here. Now, these are the different types of membrane proteins. Again, I would again refer to those two books. You have some beautiful image representations in them, in Cooper and in Karp. You can just look up Cell Biology by George Cooper and I can't, Gerald Karp, if I'm not wrong. You can look up these books. They have beautiful images and beautiful real images as well, like actual cryo-EM images of the same. So here you can see the different types of proteins that exist in a on the surface of a cell or in the cell membrane, and that allows for transport or that allows for signaling and a number of other functions. So among the different types, what you can obviously see from the first image is an integral membrane protein. When we say integral, it means that the entire protein is present within the lipid bilayer. So it is integrated, literally integrated into the membrane or the lipid bilayer. The the second type that you see here is a channel. The third here is a peripheral protein. The fourth is a globular protein, and the fifth is glycoproteins having carbohydrate chains. So what is exactly the difference between all of these? So depending on how a membrane is oriented across a plasma membrane, its nomenclature will vary. So if you have a lipid bilayer, let's consider this to be a lipid bilayer, and the protein is going across of it on both sides. If it is going across like this, that some part of the protein is embedded in the membrane, some part of it is outside, some part of it is inside, then that is considered to be a transmembrane protein. It means you are going through and across the plasma membrane. On the other hand, if a protein is just present in the space that exists between those two lipid layers, then such a protein is believed to be an integral protein. On the other hand, if a protein is such that it has been embedded in only one of the two, like if these are the two lipid bilayers and integration is only across one of the two layers, which are known as leaflets, the inner leaflet or the outer leaflet, whichever it may be, then that is believed to be a peripheral protein. It is not going through or it is not going across both of the lipid bilayers. So depending upon how a protein has been oriented, the nomenclature will vary and certainly its function will vary as well. Now talking about channel proteins. If a protein has to function as a channel, it means that it is somehow mediating the transport of something from the inside to the outside or vice versa. Or it is a transporter which performs a similar job, yet it has a major distinction. When we talk about channel proteins, they simply you can think of them like a pipe, right? A simple pipe which is giving a hollow space because the entire cell surface is say hydrophobic in nature, but there is a small gap through which some sort of a polar molecule or water or something else can come in or come out. So this sort of pipe does not have a large restriction on the direction in which this travel can happen. So whichever, say for example, if you think about an ion transporter, then the concentration of that particular ion, depending upon how it is tipped, whether the outside has a higher concentration or the inside has a higher concentration, depending on that, the transport will take place. And transport obviously always occurs from the higher concentration to the lower one. And this happens organically, it just happens across the gradient. On the other hand, when we talk about transporters, these are similar proteins in the sense that they are going across the membrane as well. But the distinction comes because of the fact that the involvement of energy is a very crucial step in transporting it. So you are transporting, you as in the cell, of course, is transporting something, some moiety, whatever it may be, across its gradient, across the ionic gradient. If you are transporting something, then that requires the expenditure of energy. And the cell is ready to do so because that transport is crucial for the cell to survive and for the cell to perform its normal function. That is why the production and utilization of ATP is done in this, just for this transport to be executed smoothly. So ion transporters versus ion channels function in this different manner. And in fact, you'll also find co-transporters like off the top of my head, one very common example is of iron glucose transporters, where one ion is used, where the transport of that ion is dependent upon transport of glucose or vice versa. So one particular ion is moved in one direction, the other moiety is moved in other direction, and therefore the expense of ATP is coupled between the two. So you can look at these examples as well. So depending upon the type of function that the protein is destined to perform, its embedding within the plasma membrane and its functioning and its orientation will also vary. You are also seeing the presence of cholesterol both in the inner or the outer leaflet. You can see glycolipids on the outer leaflet as well, because it is, as you can see, it's outside of the cell membrane. You will see the presence of these lipids, and these also are extremely crucial because the presence of such specific molecules on the surface of a cell are immunologically very relevant. They are important for certain pathogens. They're important, of course, for immune cells, like I said. If you remember the example of COVID, right? If you remember, you would see some spike proteins which were present on the surface. So these little things that you might think are irrelevant are actually very crucial when you think about a physiological system at large. So the surface of a membrane is very, very important. And like another example that I can give is of, if you have heard about it, there is a technique known as FACS, FACS, which is a method of sorting cells. So you have a mixture of different types of cells and you want to sort them. That sorting is done on the basis of what is present on the surface of the cell. So membrane composition in that regard, or rather the presence of some moieties on this, when I say moiety, it means any entity on the surface of the cell, is very crucial in segregating it into different parts. So things like this, very minor when you think about it just in this context, are very interesting when you want to perform some landmark experiments. So yeah, we talked about the membrane, and this particular model, if I did not mention it already, is known as the fluid mosaic model. Why such a name? It means that there is fluidity in the membrane. When we say fluidity, it means, obviously means that it is not rigid. This is because there is movement in the membrane. Each of these lipids that we talked about, like those two heads and that those two tails and that hydrophilic head, these individual lipids are capable of moving across that same surface. Like if we see here, this particular surface in that one particular leaflet, these lipids are constantly in motion. If you remember the video that was there in the lecture as well, a little video at the end, you could see such rapid movement occurring among the lipids. They are capable of this horizontal movement. They are capable of, very rarely, but capable of flipping, flopping, and they can move about. So there is a lot of fluidity. On the other hand, if you like in the a large number of houses, particularly in previous times, there were a large number of houses whose flooring had a mosaic flooring. So if you're familiar with it, it is usually like a yellowish background and that has little colorful stones that are embedded in it. That is what the cell membrane would look like if we just observed it from the exterior. It would look as if there is a whole background of lipids, and within it you will see little membrane proteins that are embedded here and there. So that is the appearance that it looked like under the microscope. I don't think I have the image here, but I can include it maybe in the next class, or you can look it up, where they first observe that the cell membrane looks like this using SEMS, scanning electron microscopy, which looked at the cell surface. It looked like it's a mosaic, and the fluid nature of the membrane gave rise to this theory. However, it has been updated to some extent, and now there is an updated fluid mosaic model, which I'll describe here. So, first of all, the mobility of many membrane proteins is restricted in contrast with the free movement in the bilayer that was initially proposed in the FMM, or the fluid mosaic model. So, when I talked about the fluid nature, it was not only with respect to lipids, but also the proteins which were believed to just freely move about. But that would be very detrimental for the cell, because proteins have been placed or localized at a certain region inside the cell because they are destined to perform a certain type of function. Like I said, there is a railway track that has been put in place inside the cell to make sure that whatever is every protein carries an address, and that protein is getting localized to its correct address and not somewhere else in the cell, because that could lead to again a large number of disorders. In biology, you need to understand that while your body has a large number of checkpoint mechanisms, your immune system being the major one, any major thing that goes wrong is going to lead to disease. Some of them less severe, some of them more severe. So the correct positioning and the correct occurrence of a large number of events are why so many of us, majority of the population across the world, is healthy. So yeah, a lot of credit to the work that your body is doing. Now, coming back here, so the mobility of membranes, like I said, is restricted in the bilayer. They are composed of some distinct domains with important structural and functional roles. So while the previous belief was that the entire surface of a cell is just um, like a space for the lipids to move about, it's not exactly like that. There are regions, distinct regions or distinct domains within there that have some role, some structural or some functional role. Some plasma membrane proteins are in fact associated with the underlying cortical cytoskeleton made up of proteins like spectrin and actin. If you remember that immunofluorescence image, there was a red lining along the outside of the cell or close to the membrane of the cell, which was a staining for actin itself. So membrane or lipids at the membrane are often associated or are connected to the actin that is lying right underneath, and that is why the fluidity of the cell is not as relaxed as one might think. So yeah, and that restricts mobility. It also can act as barriers to restrict the mobility of some other proteins. Then we have cholesterol that we just talked about, sphingomyelin and other glycolipids that can form these little structures which are believed to be known as lipid rafts. So if you see here in the image, we'll see these little portions, which are sort of orange, blue, pink, etc. These are known as, when you think of a raft, it is like a imagine a lake or any sort of water body on which you have a little raft on which people are floating. So bringing that analogy to a cell, what it looks like is that if the cell, if the membrane lipid is sort of like a sea of lipids, within that there are certain regions which are not as free, which are little surfaces over which some particular proteins have been localized so that they are always in close function. And while it might seem like a very pointless exercise for you and me in the first go, it has very far reaching consequences. For example, for some particular signaling path, before you have a receptor and a co-receptor or some other mediator protein which are all localized on the same raft, and it might be very, very crucial for all of them to be close together for that signaling pathway to be activated. So and that might be a survival essential pathway as well. So what you see are lipid tiny little rafts on the surface, and these are usually very rich in entities like cholesterol and sphingomyelin, as has been mentioned. And apart from that, they are also often enriched in GPI anchored proteins, certain transmembrane proteins, etc., that are involved in specific functions like signaling, endocytosis, and movement. Endocytosis is basically engulfing something. Endo means inside. Cytosis is well, by cells. It's like you are engulfing something and you are bringing it into the cell. The process of throwing something outside of the cell is exocytosis, where something that needs to be removed or cleared out from the cell would be exocytosed by the cell itself. So this process is often also dependent upon, not dependent upon, but often supported by the presence of such lipid rafts on the cell surface. Then come caveoli. So they are a subtype or a subset of these lipid rafts, and this involves a certain membrane protein known as caveolin, which interacts with a cytoplasmic protein, that is cavin, and cholesterol invaginations, and it is involved in this movement as well. So this is just an updated or a more evolved rendition of the fluid mosaic model. This was actually quite recent. I think just 10 years ago, it was in 2014 or 2015 that this version of the fluid mosaic model has actually been published. Um, Next, coming to the bioenergetics and metabolism. So already you have understood that the membrane self, like when we looked at the organelles of a cell, we saw that there is mitochondria, there were lysosomes involved, and of course, if the cell is taking up something inside this inside its system, which is by the process that I just discussed, that is endocytosis, then the cell will utilize it in some way or the other and will try to obtain energy from the same. So the initial breakdown, and this breakdown is by something we again discussed very recently, which was enzymes, right? So enzymatic breakdown of this food materials, whatever has been taken up, usually occurs in the, if you're thinking of the organism at large, then it occurs inside the intestine, because complicated large food particles are broken down into simpler molecules, and then those will be digested by individual cells. So that digestion takes place inside the digestive bags that I mentioned, which is the lysosome, and they break it down into the simpler units, monomeric units, like amino acids, sugars, fatty acids, and cholesterol. And these enter like these small organic molecules that are produced, they enter the cytosol, they get oxidized. Like, for example, glucose is converted to pyruvate by the process of glycolysis, which also involves the production of ATP and NADH. Then this pyruvate can then go from the cytosol to the energy center of the cell, which is the mitochondria. There it is converted to CO2 along with acetyl coenzyme A. And this acetyl coenzyme A is the activated carrier molecule that is present throughout this entire metabolic process by something called as oxidative decarboxylation. Here as well, CO2 and NADH are produced. And this acetyl coenzyme A can be easily transferred through high energy linkage to other molecules as well. Then this is transferred to, one example of such is the transfer to oxaloacetate once it enters into citric acid cycle. So like if you look at the pathway, food has been taken up, food has been digested, glucose has been produced, glucose is converted, glucose undergoes glycolysis, which means the breakdown of glucose in the cytosol. From the cytosol, acetyl coenzyme A, pyruvate, CO2, NADH, all of these are produced. From here, the acetyl coenzyme A goes and it is transferred. The acetyl group is transferred to oxaloacetate, and that forms citrate. And this citrate can then enter the citric acid cycle, or what you commonly know as the Krebs cycle, which has a number of steps. Not something you need to like you need to go for rote learning for, but yeah, it has some specific enzymes, again explaining the specificity of those enzymes along with certain cofactors which are crucial for those enzymes to actually function. And from the citric acid cycle, oxaloacetate is regenerated, while you'll get ATP, 1 FADH2 and 3 NADH, which are formed in one cycle of the or one turn of the cycle. From here, from these energy molecules, electrons are transferred into the ETC, which is the electron transport chain, and this is present in the inner mitochondrial membrane. And this transport occurs to molecular oxygen, and that leads to the formation of ATP molecules by the final process of oxidative phosphorylation. So all of these things are happening just to give you ATP. Seems like such a tedious job, but that is how crucial it is, and that is how it has evolved in the past several thousand years. So, sorry, I've been speaking for a long time. Again, any doubts at any point, please interrupt me. Uh, yeah. So this is kind of a consolidated picture of the same thing that I said. So you have fatty acids and pyruvate, which are this is the mitochondria. So from the cytosol, all of this is happening. So it enters into the citric acid cycle. Acetyl coenzyme enters here. It produces these electron carriers, and oxaloacetate is again recirculated into the cycle. Electrons from these will enter into the ETCs, or electron transport chains, and they will be added to O2 or oxygen to form water. While on the other hand, this water that is formed, from there you will get H plus ions, which are being transported across the mitochondrial membrane, inner mitochondrial membrane, into the matrix, and into the intermembrane space, and that proton gradient is being utilized for the production of ATP by one particular beautiful enzyme, one of the most wondrous enzymes that you can find inside the system, because it is performing that extremely crucial job of producing energy inside the cell. Yeah, excuse me. So the F1 FO ATPase, you will find it written as F1 F0 also in a large number of places. There is some debate about it, but as far as I know, it is F1 FO ATPase. So like the name indicates, it has two easily distinguishable domains. You'll have an FO domain, or F0 domain as one may call it, that is embedded in the mitochondrial membrane. And on the other hand, you have the F1, which is exposed towards the cytoplasm, towards the outside, basically. So it is what you see on the right is it's a experimentally determined structure of this exact protein. You have a much simplified version, I think, already present in the lecture, or I think you'll find it here as well. Yeah, this is the simplified structure of the same protein. But I think this is a lot more interesting when you look at the experimentally determined structure of the same, because it shows how much more complicated the actual structure is. So you'll find different colored representation of each of the different protein chains or polypeptide chains, and you can see that it is entirely made up of helical structures, right? These are alpha helices, which, like I already discussed, are the secondary structures of a protein. So you can see that there are very distinct domains. Like, for example, the head part, the steering sort of a part, which is in the F1 domain, has three alpha and three beta entities, or three beta domains. And the alpha beta interface is the point of, or is the region where catalysis actually occurs. So between the A and B domains, you can find the conversion of ADP, which is adenosine diphosphate, to adenosine triphosphate, or which is ATP, because of some conformational change that is occurring. Now, when we think about just like globular structures like this, it feels like, where is the conformation change at all occurring? But when you think about structures like this, where you can see that it's just made up of helices, you can imagine that each domain is made up of individual helices like this, and their orientation, a shift in their orientation, can lead to very far-reaching consequences. So when ATP is produced, it is released because, again, of a conformation change. So this knob structure is at the top. You have the gamma epsilon moieties, which are linking these two domains. You have an external B2 structure, which is connecting the top of the F1 domain with the bottom of the H0 domain, and it is membrane embedded as well. And the remaining C10 to 15 is localized inside the membrane. It is a membrane embedded region. So the F1 FO ATPase looks like this. The interconversion between, not interconversion, the conformational change between each of these domains is what is leading to the final production of ATP in the long run. Uh, so yeah, that was the summation of the metabolism part. Then came the idea of understanding the genetic material of the cell. We have talked about the nucleus. This will not be very long, I think. Yeah, it's just 3 slides and then we can get to questions. So yeah, understanding what is the genetic material of the cell. Like today we understand that it is, just a second. Today we understand that it is DNA, which is the genetic material of the cell. But if you ever want to understand how that discovery was done, it was not as simple as you think of it, but instead took a number of very landmark experiments, simple but very landmark experiments, that all these scientists did to finally arrive at this conclusion. Some of those were obviously mentioned in the lecture, like there was the one carried out by Avery, MacLeod, and McCarthy, where they used streptococcus and the different strains of streptococcus, one of which was pathogenic, one which was non-pathogenic, and based on the coating, they tried different combinations and they understood what is the correct genetic material of a cell. So, yeah. So here you see that they looked at all of the different options, which was RNA, protein, DNA, carbohydrate, and lipids, and they found that the transforming principle, which is what is converting a non-virulent strain or a non-pathogenic strain to a pathogenic strain, is being provided by the DNA component of these cells. So DNA was therefore considered to be the transforming principle, or it was considered to be the genetic material. After this initial discovery, there were n number of papers, only some of which have been highlighted here. For example, the one, of course, very famous scientists Watson and Crick, who gave the structure of DNA. But you definitely must credit Rosalind Franklin, who was involved in the initial experiment that actually revealed what the structure of the DNA was. The analysis part was, of course, taken care of by Watson and Crick. So they provided the basic structure, or they provided the analysis of the basic structure of DNA, and they showed how it had two helical chains coiled around one axis. They are right-handed helices. They run in opposite direction, which means they're anti-parallel. They have a phosphodiester group that joins the furanose residue with 3' 5' linkages. Bases are always on the inside. Phosphate is on the outside, forming the covering. There is a repetition of residues every 3.4 angstroms. The turn, like the basic, like if you think about if somebody has to construct DNA by themselves by hand, they'd be able to do so. That is how extensively this study was done. What you need to keep in mind is that because this was a very early experiment done very, very long time ago, sorry, I don't remember the year, but the technique that was used back in those days was X-ray crystallography, which is a method of simply, if I put it very simply, it is just crystallizing a protein. When you say crystallizing, it means you are freezing the protein in one particular conformation. In this case, it was not protein, it was a nucleic acid, which is DNA. So it was just made stagnant in the condition that it was. It was converted into a crystal, and then X-ray was used to, it was passed through the crystal, and then the way in which that X-ray was deflected because of the electrons that are present in the biological molecule that you're studying, they could detect how the actual structure is. So it's basically dependent on the principle of diffraction, X-ray diffraction technique. So that is how initially proteins were studied. Apart from this, there is another technique known as NMR, which is nuclear magnetic resonance. That is a more while it is not as high resolution, when I say resolution, the accuracy with which you can identify the subtle differences between each of the atoms of a particular biological molecule, it does not have that higher resolution, but it allows you to keep the proteins in their normal, when I say normal, I mean physiological condition, that they can move about. You're not freezing them in one condition. But NMR also has a number of limitations. Only certain weight of proteins can be studied. They must be soluble. So every technique has its own shortcomings. What is being used now in this day and age to study proteins, it's still an up and coming technique, but it's one of the most brilliant ones you'll ever see, is cryo-electron microscopy, which I mentioned previously. So you are studying those proteins in their native function, allowing them to perform. Like, for example, if you think about F1 F4 ATPase, right? It is moving. It's like a little knob that is rotating like this, and it is releasing ATP continuously. So you can capture each of those different positions. You can understand how that protein is moving. So all of this has happened now, and I'm sure in the future we'll be able to record videos of what is happening inside the cell, because right now we are taking images and we are just forming them into a movie. So everything is possible. It's just a matter of time. Coming back to what Watson and Crick did, they just got the data that initially was published, not published, but was the work of Dr. Rosalind Franklin, where she had the diffraction pattern of DNA. I'm sorry, I don't have that image as well. You'll find it very easily. It was known as photo 51, which showed that there was this sort of strange pattern that can only be brought about by a structure which is helical. So from there, they understood, or rather they decoded each and every aspect of DNA that we just talked about. They showed what is the gap between each of those units, how repeating they are, how frequently repeating they are, what is the direction in which it is arranged, how the bases are present, and how the outside is arranged in this manner that DNA can survive in an aqueous environment. Then how the bases are perpendicular to the axis, the bonds that are present between the bases, which is between purines and pyrimidines, A with T, G with C, etc. All of that. There was also a very landmark paper by Stokes and Wilson, which was about the molecular structure. They gave the X-ray photograph. They took the X-ray photograph of the DNA, and they gave the pitch, diameter, number of nucleotides, the presence of bases in the central region of the helix. So once one landmark paper has been published, such scientists were able to find more and more about the DNA today. Well, we know a lot about DNA today, but there definitely is still a lot more to learn. For example, if I put it very directly, then the Y chromosome was sequenced last year, if I'm not wrong, 2023. So imagine the amount of information that would come out of just that single chromosome being decoded. So much more would be studied. Why is it smaller than all of the other chromosomes? How is it arranged? How is its inheritance different? So there is always more to learn, is the point that I'm trying to make. So all of these studies, while they were primitive, they have been very crucial in understanding of the DNA. Then was a study by Franklin and Gosling also took the same photo of the same X-ray diffraction photo, and they are further provided more information, and it was consistent with what had previously been submitted by all of these scientists. And apart from that, here you can see a more detailed description of the way in which this binding actually occurs between AT as well as GC. And like you can see, the binding is always between a purine and a pyrimidine, which are the two types of nitrogenous bases that make up DNA. So you have a nitrogenous base, you have a ribose sugar, and you have a phosphate molecule. So each of these are arranged in a way such that you get a phosphodiester bond. So this is the type of bonding or the type of base pairing that is observed physiologically, and this was provided by Shargov, who was another scientist, and therefore this is part of Shargov's rules as well, that only A and T can pair with each other, G and C can pair with each other under normal circumstances. There are, of course, very rare exceptions, but they do exist. On the right, what you see is another experiment. Like I think it's important to understand experiments because all the information that we get today, we take it very lightly in a very matter of fact way. But the fact that very crucial experiments have led to those conclusions is something that I think we all need to appreciate. So the experiment that was done to understand what exactly happens, like after they initially discovered that DNA structure is in that particular manner, and they saw that there is an anti-parallel nature in which DNA is present, they understood that it would have some significance in the way that DNA replicates, and indeed they were correct. So the experiment that was performed was by Meselson and Stahl, one of the most beautiful experiments that you can, so simple and yet so beautiful. So they actually understood how DNA replicates semi-conservatively. So they grew bacteria in two different types of media, one with a regular isotope, which is said to be, which is said to be the cold medium, versus one which is in a heavy medium, which is said to be a hot medium. So this hot and cold comes from the radioactive nature of each of those. So when a particular isotope is radioactive, it is said to be hot, versus when it is the regular isotope, then it is said to be cold. So they were grown in two different types of media, and they were sampled at the initial time and sampled after 20 minutes and 40 minutes and so on and so forth. So what exactly you see, if it was conservative in nature, like there are three possibilities that DNA replication could be conservative, semi. Uh, Whoever is trying to mute me, please don't. The lecture is being recorded, so it would not be very helpful for you. Yeah. So if you have a question, you can ask. Uh, Coming back here, we were looking at the experiment by Meselson and Stahl. So you can see that there is a that semi-conservative nature of the DNA replication was indeed observed in this case, where they saw that after 2 generations, the weight of DNA was intermediate. It was not completely heavy, it was not completely light, and they found chains of both types. Whereas after, sorry, this was in the first generation. However, after the second generation, they could find a mixture where it was either completely light or it was a intermediate weight. So they could see that the semi-conservative nature was being conserved over several generations. So that was the nature in which DNA replication was carried out. And this will be the final slide for the recap. We'll just go over some questions, and nobody has interrupted me yet, but please do if you have any questions at all. So apart from these basic studies, what else was discussed here was the central dogma. So we have already discussed it, and we have tried to correlate it with the arrangement of the organelles inside the cell, that how the nucleus, the endoplasmic reticulum, and the cytosol have been or are arranged in a manner that allows each of these processes to occur sequentially. So the dogma basically means a hypothesis or an idea, let me put it that way. So the dogma was about the basic sequence of events that is carried out in a cell. So first of all, what you have is the genetic material, which is the DNA. So that DNA undergoes replication. After replication, it is being transcribed from DNA to RNA, and from there, it is being translated from RNA into protein. So if you see, there is a unidirectionality that was initially believed to be the case for any and every cell. But of course, the experiments by Temin, they showed that there is a DNA provirus sort of a stage. You have already seen all of that in the lectures, but basically what he shed light on was the idea that no, it is not that unidirectional a process, but instead there are exceptions to this as well. You will see that there is reverse transcriptase, an enzyme which is extensively found in viruses, retroviruses, and it is extremely it was a landmark discovery again, because it could be seen for the first time that there is a reversal in the directionality of the central dogma. So for the reverse transcriptase, the RNA genome of viruses is being converted into cDNA or is being converted into DNA, and then that DNA is being used to propagate or is getting integrated into the host genome, or a number of things are happening. If you are interested, you can look for reverse transcriptase in mammalian systems, let's say humans. You will find examples of the same. So that would be an interesting discovery. Unless somebody already knows, you can share that. So yeah, so that is what you see here. Like the retroviral life cycle has been represented here. You have the retrovirus, which is binding to the cell surface. Again, like I said, the membrane composition is very, very crucial. It is recognizing certain proteins on the cell membrane and is utilizing that as a host, fusing with the cell, inserting its viral RNA genome, which is getting reverse transcribed into viral DNA. Yeah, into viral DNA, and then that viral DNA can enter the host nucleus, which is again where the nuclear pores are coming into use. That is getting integrated into the host genome, transcribed, translated, and new viral particles can be formed and they can be released out of the cell. So this initial process is endocytosis, the later process is exocytosis, and the middle is the host, is the life cycle inside the host, where reverse transcription is playing such a huge role. So just an observation of other living systems, pre-existing living systems, can lead to such extensively beautiful discoveries. And the idea of reverse transcription is something that we routinely use in today's day and age, particularly for something that you all might be quite familiar with, which is qPCR. So the RNA that we isolate from whatever cells we are studying may be a patient tissue sample or might just be cell lines. So we whatever RNA we isolate from there, we reverse transcribe that to produce cDNA or complementary DNA. And then that complementary DNA is used for carrying out qPCR or quantitative PCR to measure what is the expression of that particular gene at the mRNA level inside the cells. So just a simple discovery, extensive use in biological research. So again, do not never negate your results. It can lead to wonderful discoveries. And after this, I think the other two topics that were discussed were transcription and translation. I'm not getting into details of those because a recap of those would be very extensive. But again, if you have any doubts, you can go ahead and ask. But the basic idea is, of course, converting the mRNA into converting the genetic material or the gene into the mRNA in the steps of transcription, where you'll have initiation, elongation, and termination as the three major steps. The molecular machinery, which is in charge of this particular process, is of course the RNA polymerase, or to put it a little more elaborately, it is the DNA dependent RNA polymerase. I have said it, I think, some other session before, but these polymerases can be thought of as sort of a copy machine, where but a slightly dysfunctional copy machine, because it takes one type of input, produces different type of output. So it takes the DNA as your input, produces the mRNA as the output, whereas the enzyme involved in replication, it will take it is otherwise known as a DNA dependent DNA polymerase. So it takes DNA as the input, gives DNA as the output. On the other hand, the ribosome, which is not just a regular protein, it is also an enzyme. So the ribosome takes the mRNA as the input, and it produces, or it reads the mRNA and translates that mRNA into the corresponding protein. It takes each of the three codons converts that into one particular amino acid and joins all of those amino acids to form a polypeptide, which eventually forms a protein. So for translation as well, the basic steps are basically they are similar. Um, you will have the binding of the tRNA with its cognate amino acid, linking between each of those amino acids by the process by an enzyme which is known as peptidyl transferase, which is also present inside the ribosome itself. It joins each of those amino acids and elongates the amino acid chain or the polypeptide chain. And after complete elongation has been carried out, the process of translation will then be concluded or terminated. So that is the entire process which is carried out at large. So since this has already, I think you will find a lot of lectures in detail about it. But again, if you need details, please let me know. But that basically sums up the topics that were a part of week one. Now I don't have a large number of questions. I just have a very few. But yeah, we can go through them and we can look at another set just for reference, a slightly older question paper from this course itself, if that is helpful. So we'll look at these first. So first of all, the question is about how gram-positive bacteria, they are stained purple with crystal violet because of what reason? So like I initially mentioned, that gram staining is the procedure via which gram-positive and gram-negative bacteria are distinguished. And that distinction is brought about because of the type of composition of the outer layers of those bacterial cells. So anything that is gram positive gets stained purple, whereas anything that is gram negative gets stained in the reddish sort of color. So the reason that gram positive bacteria, they get stained purple is because they have a thick peptidoglycan layer and they do not have any outer membrane. So nothing is retained inside the cell and therefore they get stained in the purple color. The second question is about which statement does not hold true for the nucleolus. So we talked about the nuclear compartment and we said that how the nucleus, the interior parts of the nucleus are not membrane bound, but there is, while there might not be a very distinct segregation when we look at it at large, again, this has been studied at the microscopic level and there are, at the molecular level as well, there are a lot of differences, but there is no lipid bilayer or there is no membranous segregation between these two compartments, but the nucleolus and the nucleoplasm are segregated from each other. So the statements that have been mentioned are that A, it is a dense body present inside the nucleus, which is true. It is the site of rRNA synthesis. Yes. So this is something that you can look into if you're interested. You can look at where tRNA, mRNA, and rRNA are synthesized inside the cell. In fact, you can also see where assembly of ribosomes is taking place, because ribosome is a macromolecular machinery that is made up of these two things exclusively. It is made up of RNA and it is made up of ribonucleoproteins or RNPs. So the proteins are of course synthesized in the cytosol, but the RNA is being synthesized in this region. So different types of rRNA also present. So this assembly, how does it occur? Where does it occur? You can look it up if you're interested, or we can discuss this in the next class as well. So it is indeed a site for ribosome assembly as well. The only statement that does not hold true is that it is bound by a thin membrane, which is of course not the case. Yeah, the third question is about the plasma membrane or the cell membrane. It is termed as selectively permeable because A, it allows the free diffusion of gases, hydrophobic and small uncharged polar molecules such as water, and it also contains transporters to selectively transport large polar uncharged molecules such as glucose, which is basically the function that we are most familiar with. And it is, of course, the correct option as well, as I've highlighted. The other options we can look at as well. The only way to cross the cell membrane is via the transporter proteins, which is not true. If you remember correctly, I said that it is a fluid mosaic model, right? It is fluid in terms of movement, and it is also fluid because it allows, it's not a wall, it's a membrane. Whenever we say membrane, the difference between a wall and a membrane, a cell wall is also present in plant cells, right? But the difference between these two structures is that the membrane is more relaxed. The membrane has some scope of entry and exit of certain molecules, whereas a wall is extremely rigid. So while it provides mechanical strength, it does not allow for very easy exchange of anything across these across that boundary. So in case of plants, there are other sort of structures known as plasmodesmata, which are openings in the cell wall as well, that allows for transport, intercellular transport, that is transport among cells. But in case of animals, we do not have any sort of cell wall under normal circumstances. There are exceptions to this. But instead, we have membranes that are semi-permeable, or in fact, to be more accurate, they are selectively permeable. That means these membranes are responsible for allowing the free diffusion of a number of things, some gases, water, ions, etc. And it needs certain help, or it needs ATP dependent, energy dependent help of certain transporters to transport large molecules, which are, for example, glucose. Like I mentioned, co-transporters are present in a large number of cells. So yeah, that is the correct answer. And it completely restricts the movement of large polar uncharged molecules across itself. Of course, it's not true. It has put in mechanisms in place which will allow for this transport, and that is via these specific membrane proteins. Um, Question 4 is for picking the correct statement. A, guanine is a purine base, thymine is a purine base, cytosine is a purine base, adenine is a pyrimidine base. So you should, there is no easy way about it, but you must remember the at least the difference between purines and pyrimidines, because the structures are a little complicated and they're not easy to remember. But what you can keep in mind, and how I personally do it as well, is that you write somewhere that A bonds to T, double bonds to T, G triple bonds to C. This is not the chemistry bonding, but yeah, it's three linkages between the between G and C, two linkages or two bonds between A and T. Now on the left, what you have A as well as G, these are your purines, that is adenine and guanine. What you have on the right, which is T and C, those are your pyrimidines. T is thymine, C is cytosine, and of course, in case of RNA, it would be uracil instead of thymine. So these two are your pyrimidines. So that is how you remember the distinction between the two. So among these, the correct statement, if we have to look at, purines were A and G. So among them, the first statement obviously becomes correct. That is, guanine is a purine base. The rest are not true. Uh, Question 5 is for Shargaf. If you remember the place where I showed the bond between those, between the bases, nitrogenous bases, I mentioned Shargaf, and Shargaf's rule states which of the following. The first is ratio of amounts of A to G and T to C are close to unity in DNA. When we say close to unity, it simply means, like, what does the statement mean? It means the ratio of amounts of these bases. That means if A and A and T are equal, then that means A by T equals 1, or G by C equals 1. So it basically means that these are equal in amount. It's just a different way of putting across the same thing. So based on Shargaf's rule, it should be A and T, and it should be G as well as C. So depending on that, the correct option would be option C, where the ratio of amounts of A to T and G to C are close to unity in DNA. Coming to question 6, which of the following organisms belongs to the prokaryotes? So you have yeast, animal cells, archaea, and protista. So while we have not discussed the like all of the kingdoms and everything in detail, but basically, if you're just simply asked about prokaryotes, then of course archaea or archaebacteria, they are part of the prokaryotes. Yeast are not. Yeast are unicellular eukaryotes. Animal cells definitely are not. Neither are protista. So the correct answer to this would be archaea. Coming to question 7, which of the following is true regarding carbon atoms? So carbon forms unstable carbon-carbon single bond. Certainly not true. It can form 4 covalent bonds. It is true. It is tetravalent in nature because of the free electrons or valence electrons that it has. There is an obvious upper limit to the size of the rings or chains that are formed by carbon atoms. Certainly not. Upper limit is quite a difficult way to put it across because it is tetravalent in nature, but it's not really an upper limit. And fourth is all of the above. So because option C is not entirely correct, option B would be correct in this regard. Question 8 is about which of the following allows the ions to diffuse down the concentration gradient. Ion transporters, ion channels, lipid bilayers, all of the above. So because we are just talking about an energy independent method of transportation that is just dependent upon diffusion, and that diffusion depends of course upon the concentration of the ion on either side. So that diffusion across the concentration gradient will occur through channels and not through transporters, which is what I initially said. So lipid bilayer would also be something that somebody might say, but it is not the norm and it is not something that will allow ions under normal circumstances to be transported. Gases can transport or diffuse across the plasma membrane, like oxygen, for example, can diffuse, but certainly not ions because they will carry a charge and that will not be able to penetrate the lipid bilayer. It would not be stable. For that, you certainly need an ion channel to help movement across the membrane. The last two questions. Let's skip a question. Yeah. So question 9 is about the bond between the nitrogenous base and the sugar group in a nucleotide. So the nucleotide structure, like I mentioned, has a nitrogenous base, it has a sugar group, and it has a phosphate moiety. The base and sugar are linked by a glycosidic linkage. When like the name itself will indicate that, right? That you have a there is some sugar moiety because it's a glycosidic bond. It will involve some sort of a sugar there. So that linkage is what links these two. And among the different phosphates that are present in the nucleotide NTP or nucleotide triphosphates, the linkage that is present would be the phosphodiester bond. So you can again go back to the structure of DNA to clarify your doubts if there are any. But what is important here in this regard is that what type of glycosidic linkage is present. It is the N-glycosidic linkage and not the O1. So that needs to be kept in mind. And finally, a very simple question that in an RNA molecule, uracil replaces which of the following bases that are found in DNA? Adenine, cytosine, thymine, or guanine. Then certainly uracil would replace thymine in case of RNA, whereas you would have thymine in case of DNA. So you can actually look at what is the difference between the two. It's not much. But yeah, that's the difference that are present. And the reason, while it has not been very concretely been elaborated anywhere, it is believed that uracil is more it has some specific functions. Let's just put it that way. It is interesting to look at, and there have been multiple hypotheses. Some say that it is stable for RNA for a single-stranded moiety. It is more stable, but then why at all did thymine come into play? So there are different approaches in this regard. But yeah, the answer to the question here is definitely thymine, which is replaced by uracil in case of RNA. So yeah, that actually sums up my presentation for now. So in case anybody has any questions, please do ask me now. I'd be happy to answer them. And anything that I explained, if it is not clear, please go ahead and you can ask me.
How how can we get the PPTs?
Yeah, I'll share it. You'll get the link. I'll be sharing the PPT and also the video lecture link, like the recording from today. You'll be getting that.
Okay, ma'am. Thank you.
Welcome. If you want me to include more questions, I can do that. There were just 10. I'll try to get more questions and maybe more images, which are not a part of the actual video lectures that you have. That might be helpful. And if there are any other suggestions, you can certainly get them into notice at the platform that you have on the course as well. The feedback form as well, if you want. And any doubts and queries that you may have, you can drop those um, today. If not, it's fine. You can keep your questions. We can even discuss it in the next class, or whatever you want to do, we can go ahead with that. Uh, if anyone has any more questions, please go ahead. Otherwise, I'll proceed with ending the meeting for today. Okay, if not, then thank you so much for joining. I hope it was helpful for you. I'll try to get more questions because that might be a little more beneficial. In the next week's interaction session, we'll be discussing this week's assignment because your deadline would be complete by then. And also, I'll come up with some different types of questions because that would be helpful. And we'll go through a recap of week 2 as well. So please give your suggestions. Please ask doubts if you have any. Please do not hesitate. And any help you need, please let me know. I'll be very happy to help in that regard. So thank you so much for joining. I hope you have a wonderful night. So thank you and good night.
Yeah.